Whether it is labware or culture medium, plant material or instruments used for culture in the laboratory, all are sources of contamination and need to be sterilized. The routine sterilization procedure of non-living articles, such as nutrient media, glasswares, distilled water, and instruments, is by autoclaving under steam at a pressure of 15 psi and a temperature of 121 degrees Celsius for 15 minutes. It should be noted that when autoclaving screw-cap bottles, the caps must not be closed too tightly in the autoclave so that gases can expand without the risk of explosion. An alternative method of sterilizing glass goods and instruments is by heating in an oven at 150 degrees Celsius for 3 to 4 hours. Thermolabile compounds are often sterilized either at room temperature or in cold by passing through bacteria filter. Plastic containers such as Petri dishes or plastic centrifuge tubes that cannot be heated are usually sold after sterilization with ethylene oxide gas, and they cannot be re-sterilized. Items in the transfer hood or laminar flow may be sterilized by germicidal lamps. UV lamps should not be used when people are present because the light is damaging to eyes and skin. Plants left under UV lamps will die. It is also possible to sterilize items in a microwave where necessary. Plant material which is to be cultured should be surface sterilized to remove the surface-borne microorganisms. This procedure is done in front of a laminar airflow or inside the inoculation chamber before the plant material is placed onto the culture medium. Commonly used disinfectants for plant tissue culture include calcium hypochlorite, sodium hypochlorite, hydrogen peroxide, and others. The use of two-step sterilization procedure, which involve placing the explants in a 70% ethyl alcohol solution prior to treatment with another disinfectant solution, has proven beneficial to explant sterilization. For one of the examples, commercial bleach contains about 5% sodium hypochlorite, so the use of concentration at 10 to 20% is equivalent to 0.5 to 1% of sodium hypochlorite. Adding a wetting agent such as twin 20 or 80 into the disinfectants can reduce surface tension and allow better surface contact between the plant tissues and the disinfectants. Conducting the sterilization process under vacuum can result in the removal of air bubbles and provide a more efficient sterilization process. Other procedures of aseptic technique include the following. Put all the sterilized articles such as media, instruments, and glass goods on the glass racks of the inoculation chamber or the table of airflow cabinet, and the airflow will blow the bacteria-free air over the working surface. Put on the switch of UV lamps of inoculation chamber for 1 hour before work, and remember to turn off the UV lamp before entering inside or work in the chamber. Put on the power of laminar airflow to the air blow for at least 15 minutes before work. Exposure to UV light builds up a high concentration of ozone gas, which is toxic inside the closed chamber. It is therefore healthy to enter the chamber only 15 to 30 minutes after switching off the UV lamp. The working glass tabletop of the inoculation chamber or the table of laminar airflow is swabbed with alcohol before starting work. Always wear a clean lab coat and use a mask. Clean the hands with alcohol and dry it. Always keep away the hands moistened with alcohol from the Bunsen burner. Pour alcohol in a clean coupling jar and dip all instruments into it. After that, flame the instruments using Bunsen burner or spirit lamp each time before use. Take the surface sterilized or aseptic plant material in a sterile Petri dish. Flame the neck of culture tube or flask and quickly remove the plug of glass wire before transfer the tissue onto the medium and replace the closure. Lastly, do not dip hot instruments in alcohol and don't use hot instruments for cutting or holding the plant material. This is the end of the lecture. Thank you for your attention.